Hi, this is Henry Pinchbeck of 3D LifePrints, and I'm talking today about the rapid development of phantoms for medical research and training. I'm the CEO of the company, and I work out of London. I've also got a couple of my colleagues on the call who'll be talking about their individual projects. So first of all, let me tell you a little bit about 3D LifePrints. So we're a medical 3D printing company, which means we make patient-specific medical devices. Our business model is that we embed ourselves within hospitals, and you can see on the right-hand side of the slide here are three current hospitals up in Liverpool, one in Wrightington, which is near Wigan, and one in Oxford at the Nuffield Orthopaedic Centre. We're also opening up in the next year in Leeds and in London, so watch this space. Our business operates at something that we call the point of care, which means that we deliver our services directly to the clinicians from inside the hospital. That's a really novel way to provide healthcare solutions, and we think it gives the hospital exactly what they want and also gets us right up next to the clinicians that we need to for our regulatory and design purposes. We're technology agnostic within the company. We'll use any kind of tech, well, not just 3D printers, although that's obviously a big part of what we do, but some of the solutions you'll see may well not have been 3D printed. We're multidisciplinary. We work across all the surgical disciplines, anything from orthopedics through to cardiology, craniofacial, and oncology, a really big area for us. Two of the things that we work on really tightly within the business are regulatory compliance and quality assurance, and that's very much to differentiate ourselves within the industry and make sure that we're providing a service that meets all the regulations and quality requirements. A little bit more about the products. You can see on the right-hand side here, there's a good mix of the products in this particular case. There's an anatomical model, that's the brown thing, that's the acetabula. The yellow, the two tones of yellow, is a part of that model is the tumor. You can see the tumor in the darker yellow and a region around it. And the white thing is called a cutting guide. So an anatomical model, you guess what that is, it's just a copy of the anatomy. A cutting guide is something that gives the surgeon a plane to cut along, so they're patient-specific devices again. And implants, of course, we do design, manufacture, and deliver both orthopedic and craniofacial implants. But what we'll be talking about today is the second section of our business, which isn't clinical, which is all to do with simulators and training devices. Now, these are non-patient specific devices, at least normally non-patient specific devices, and we develop them normally in collaboration with surgeons or clinicians at certain hospitals around the country and actually around the world. When we get onto one is from America. So without further ado, I'm going to pass over to the first case, which is by Camille Ribolzi, who will be telling us about one of the devices we developed in London. So Camille.
Thank you, Henry. Hi, everyone. My name is Camille. I'm working for 3D LifePrints as a biomedical engineer. I joined the company a bit more than 2 years ago, and I'm based in London. So I'm going to talk about a simulator we've developed for Evelina Hospital, and more particularly for surgeons at the Pediatric Intensive Care Unit, which is the simulator you can see on the screen. It's called the Jugular Vasculature Access Trainer because surgeons will use it to practice the cannulation of the internal jugular vein. The cannulation can be performed during a low-invasive procedure, which is guided by ultrasound, or during surgery where a patient needs the support of an ECMO machine to replace the function of their heart and lungs. In this case, both the jugular vein and the carotid are cannulated, and this simulator can be used for the training of both the low-invasive and the invasive procedure. So for this reason, the simulator is made of 3 parts. The baby main body that you can see in pink is the realistic external baby anatomy, which is made from silicone. The second part is the replaceable inner section, which contains all the vessels, muscle, and bone details. And this part can be replaced after the surgery is simulated on the model. And finally, the third part is the skin pad, which is being cut by the surgeon on the picture on the right. And this one can be replaced as often as needed because it's a low-cost part. This model that we've developed is fully ultrasoundable because the procedure can be performed so that the procedure can be performed in realistic condition. So indeed, when they practice the procedure, the surgeon try to feel the clavicle and the sternum through the skin to get some landmarks. And we also reproduce the blood circulation in this simulator, as well as the pulsatility of the carotid, in order to have realistic conditions. When they use ultrasound, they would target the vessel and cannulate them. But when the surgery is simulated, the surgeon would dissect all the tissues up to the vessels to cannulate them. In both cases, the success of the procedure is achieved if the fake blood flows from the cannulas. So the challenge of this simulator was really to select the right materials to provide the surgeon with a realistic feeling, but also the materials needed to behave realistically under imaging, in this case, ultrasound. I'm going to pass now over to my colleague David, who is going to speak about another interesting project, which is the head phantom. Thanks.
Cheers, Camille. Collins, I'm the 3D LifePrints Alderhay engineering lead, so managing our embedded 3D printing hub at the hospital. So today I'm going to give you a whistle-stop tour of a 3D printed medical imaging phantom that I've been working on for the University of Nebraska Medical School, or UNMS, as I will call them throughout the rest of this talk. So if we can move on to the next slide. For this project, the idea has been to develop and manufacture a 3D printed head phantom for clinical research that will allow researchers at UNMS to define and refine appropriate CT scan protocols that minimize radiation dose and metal artifact while also maximizing image quality. So development of the phantom is therefore centered around these aims and dictated that key features should include things like a removable orbital floor implant, which you can see here in the top couple of pictures, multiple dental fillings, and the use of 3D printed materials that act as appropriate tissue surrogates based on scanned Hounsfield unit values. So where we are up to currently, well, as you can see in pictures, and we have completed all anatomical modelling, which you will have got an insight from the first slide. And we have designed and finished printing the orbital floor implant, which again you can see in the top two pictures. The implant's been printed in titanium and has been designed to fit the phantom anatomy. In addition, dental amalgam has also been sourced to the fillings. So having completed elements, we're now working through the final and arguably trickiest part of the phantom development, which is the selection, analysis and identification of 3D printed materials that would act as appropriate tissue surrogates on the CT scanning for the following tissues. If we just move on to the next slide, actually. So these tissues are grey matter, white matter, cortical bone, tubercular bone, and dentition. So to start this part of the project, a literature review was conducted, and based on this, a number of material candidates were chosen. So samples of these were then printed, and we sent these across to UNMS, where they were scanned. And since then, I've been analysing the image data using custom scripts developed in Octave to identify the most suitable materials for the final manufacturing of the phantom. So I can quickly run through this analysis. For the analysis, the Hounsfield unit mean, range, and standard deviations for all the scanned samples were calculated and then compared to known values from the literature, as well as a SPECT calibration phantom from UNMS. So I've also converted all data into volumetric heat map representations, which I'll show you in a minute, of all scanned samples as just a way to visualize the overall HU values, with kind of bluer colors equating lower HU values, i.e. less dense tissues, and red equating to higher HU values and more dense tissues, really just a sanity check. So if you move on to the next slide. So starting with the brain material candidates, I'm slightly apprehensive about identifying the materials that would allow for grey-white matter differentiation, given their close proximity to each other in HU values. It's been possible to identify two material candidates as potentially suitable. So as we can see in the top right graph, their mean HU ranges correspond well to those found elsewhere in the literature. And although much of the data is kind of clustered around these means, as you can see in the bottom two graphs, the nature of the materials have allowed for some heterogeneity and HU value, reflecting some of that heterogeneity you'd find in brain tissue under CT. So moving on to the next slide. So yeah, this is just showing the volumetric heat maps I mentioned earlier, with blue and green colours representing some of the heterogeneity and the sample's low HU value in accordance with the low values of brain tissue. So if we skip the next one. Yeah, keep going. Cool. So yeah, looking at the trabecular candidates, several different materials for child as trabecular surrogates, detailed here, PFIF and PIA. So whilst PFIF seemed to capture the heterogeneity seen in trabecular bone under CT scanning in terms of the range of HU values found, as seen in the bottom right-hand graph, the upper end of the range is really too high for it to be considered a kind of sensible candidate. The HU mean of the sample PIA, however, although lacking that trabecular-associated heterogeneity, as seen in that bottom left graph, perform well when compared to the Nebraska spec phantom and in the literature as well, as seen in the top left and top right graph. So as such, this has been identified as a good trabecular candidate. And then if we move on, again, just some more visuals as sanity checks. We can kind of skip through this to the next candidate, which was cortical and dental candidates. So in contrast to the brain candidate materials, I was kind of more certain about the cortical bone material candidates selection prior to scanning. However, as you can see in the top right, although the mean value of PFIF was sort of in line with the kind of values you'd expect for cortical bone under scanning, there was a kind of surprisingly large clustering of pixel values right at the upper end of the HU scale in this bottom left graph, which don't really reflect the Hounsfield unit values you'd kind of expect to see in a patient. So while these higher values identify PFIF as a potential dental surrogate, as a cortical surrogate, either an alternative material should be found, or instead I'm currently thinking that it might be possible to take advantage of the partial volume effect instead. So if we move to the next slide, and again, yeah, so just with the partial volume effect, I'm thinking by printing the cortical shell with a nozzle diameter that's smaller than kind of normal and resolution, with air on one side and trabecular candidate on the other, partial volume effect might act to lower sample candidates HU value sufficiently to make it usable for cortical bone. So yeah, that concludes my Whistlestop to Order project today. Questions remain about a suitable cortical surrogate, but we can resolve this soon. And then material candidates have been identified for the other relevant tissues. So all in all, we hope to finalise the project and manufacture in the not too distant future. Thank you.
Perfect. Thanks, Dave. And we'll be taking some questions afterwards if you have them. Thank you.